Hello everyone, welcome to Get My Class. So let's solve this question which says which of the following statement is false? Option A, probes are single stranded DNA or single stranded RNA. Option B says probes can hybridize to its complementary region. Option C, probes can be labeled with biotin. Option D says none of the above. So let us understand the question. It says which of the following statement is false? So option A says probes are single stranded DNA or RNA. That is absolutely right. So basically, what are probes? The probes can be, as it is mentioned, they can be single stranded DNA or single stranded RNA. Basically, these are complementary for the gene to be detected, for the gene or you can say sequence to be detected. So if we want to detect a particular gene or a sequence, what do? We create probe for it, which is complementary to this particular gene. Okay, suppose this is, you have a big stretch of DNA and you have a stretch of your, you want to know where your gene of interest is. Okay, but you know it's complementary, you know the sequence of a particular gene, but you don't know the position. So what do? You create a complementary. Suppose this is DNA, this is DNA, and we have to create complementary strand for this. So either you can create complementary DNA or complementary RNA. Yes. So let us see. Suppose this is a complementary DNA. So it will have certain, of course, nucleotides. It is made up of nucleotides, right? So it will have its own nitrogen base. And this will also have its own nitrogen base. Okay, suppose, but you don't know where is the exact sequence present. Okay, where is that particular gene present? You don't know. So what do? We use certain probes and then we will radio label them. That means we will label them with certain components like ethylium bromide or anything which will help us to detect where our probe is. So wherever our probe is, that particular area, you can find your gene of interest. Suppose here it comes and attaches. Okay, suppose here it comes and attaches. Okay, attaches. Now we know that this is the region where our gene of interest is there. So this is the part of the DNA. Okay, and now how do we detect? So as I told you, these will be labeled. These will be labeled with certain chemicals or it is a radioactive components, which will show the result, like showing either change in color or glow in UV light, etc. So that we will know where our probe is. So wherever our probe is, there our sequence will be there on the particular part of the DNA or even RNA. Okay, so they hybridize with each other. The probes will hybridize with the wherever complementary part is there on the particular genetic material, it will hybridize, and this will glow under suitable condition, or this will give color or either glow, so we can detect it because of the labeling part. Okay, so option A, that is absolutely right. Now option B says probes can hybridize to its complementary region. That is also correct. Yes, they can hybridize, and that's how we can detect the sequence. Then option C says probes can be labeled with biotin. Okay, now what is biotin? This is a kind of a protein. Okay, this is a kind of a protein which can be detected by adding its complementary antibodies. Okay, so this can be used as a labeling. Okay, that means this can be introduced as labeled. This can be introduced within the probes what we are providing. That means we can label our probes with biotin as well, because biotin is a protein which will show the result by whenever they get complemented with their respective antibodies. Okay, so this is also correct. So option D says none of the above. Yes, that is the answer, because none of the statements are incorrect. All are correct. Hence, option D is the correct answer. So let's conclude. What is the answer? The answer is option B. That means none of the statements are incorrect. All of the statements are correct. I hope you understood the explanation. Thank you.